Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I am Naglaa Elmongy, Professor of Dermatology, Cairo University, and our video today will be about the effect of vagus nerve stimulation on inflammation. For a long time, the common belief was that the nervous system and the immune system were separate in terms of both structure and function. However, it's now clear that these systems are strongly connected and interact with each other, creating a continuous neuro-immune communication throughout the entire body. And from approximately 20 years ago, researchers made a significant finding indicating that the vagus nerve plays a role in regulating levels of pro-inflammatory cytokines and inflammation. Subsequent investigations through various preclinical models have demonstrated that stimulating the vagus nerve leads to a beneficial reduction in pro-inflammatory cytokines across several inflammatory conditions. As a result, the equilibrium between pro-inflammatory and anti-inflammatory cytokines has become the primary indicator of a favorable result from the stimulation of the vagus nerve. It was shown that the vagus nerve plays a role in modulating systemic cytokine levels, including tumor necrosis factor, and that applying electrical stimulation to the vagus nerve decreases cytokine levels induced by endotoxins. However, cytokine concentrations represent only one aspect of an effective immune response. It was found that vagus nerve stimulation governs inflammation through supplementary mechanisms as well. These discoveries highlighted the vital interconnection between the nervous and immune systems in regulating inflammation, which led to the birth of the field of bioelectronic medicine. In response to an immune stimulus, inflammation is initiated through the recruitment and activation of leukocytes, accompanied by significant mediators known as cytokines. These cytokines, including tumor necrosis factor, constitute a diverse group of signaling molecules that play a vital role in orchestrating the inflammatory action, leading to both individual and cooperative impacts that can induce either pro-inflammatory or anti-inflammatory outcomes. Maintaining equilibrium between pro-inflammatory and anti-inflammatory cytokines is of vital importance under specific circumstances like inflammatory bowel disease and rheumatoid arthritis. This equilibrium bends towards persistent pro-inflammatory environment, mediating inflammation and cell death. A physiological mechanism known as the inflammatory reflex, facilitated by the vagus nerve, regulates the levels of pro-inflammatory cytokines and the inflammatory response. The efferent arm of the inflammatory reflex begins in the brainstem nucleus, the dorsal motor nucleus of the vagus. Neurons originating from this nucleus travel along the vagus nerve and reach the celiac superior mesenteric ganglion complex, where they interact with the splenic nerve. Within the spleen parenchyma, the splenic nerve discharges norepinephrine, which triggers the secretion of acetylcholine from a specific subset of T cells that express the enzyme choline acetyltransferase. Subsequently, acetylcholine exerts its effect by interacting with alpha-7-nicotine acetylcholine receptors present on macrophages, leading to the inhibition of the synthesis and release of tumor necrosis factor and various other pro-inflammatory cytokines. This intrinsic reflex can be augmented by external triggers such as electrical stimulation, which has demonstrated efficacy in preclinical models of numerous inflammatory disorders. So this understanding has been applied to achieve positive outcomes in clinical trials involving the use of electrical or bioelectronic vagus nerve stimulation for individuals with inflammatory bowel disease and rheumatoid arthritis. The selection of these conditions for initial assessment and validation was because of the notable impact of vagus nerve stimulation on serum tumor necrosis factor levels, which is a basic molecular agent in inflammation and disease progression in these conditions. Preclinical investigations into the effectiveness of vagus nerve stimulation in managing inflammation have primarily centered around the cytokine reaction to stimulation. However, cytokines represent just one essential aspect of the immune response, and we have to notice that vagus nerve stimulation plays a more expansive role in modulating the immune system. Here is an important study about the effect of vagus nerve stimulation on inflammation, which was published in May 2022, and its title is Vagus Nerve Stimulation Promotes Resolution of Inflammation by a Mechanism That Involves ALOX15, which is arachidonate 15 lipoxygenase, and requires the alpha 7 nicotinic acetylcholine receptor subunit. In this study, the research group described the impact of vagus nerve stimulation on inflammation resolution using a peritonitis model induced by Zemuzan, which is a model commonly used to study systemic inflammatory response syndrome and multiple organ dysfunction syndrome. They showed that how vagus nerve stimulation significantly shortened the duration required for inflammation to subside. Also, they show that the vagus nerve stimulation facilitated both what's called efferocytosis, which is a process by which exhausted neutrophils are engulfed by macrophages, and the development of a pro-resolving lipid profile through the activation of the alpha 7 nicotinic acetylcholine receptor. Part of this effect was also dependent on what's called ALOX15 or arachidonic lipoxygenase 15, a key enzyme in the creation of specialized pro-resolving mediators. And we have to know that among the predominant leukocytes in the circulation are neutrophils, which are short-lived granulocytes, and these neutrophils represent one of the earliest leukocytic types to be recruited to the infection site. The central function of these neutrophils in infection site involves the discharge of neutrophil extracellular traps or nets consisting of their own DNA and the extracellular matrix. These nets function to capture the invading pathogens, which are later engulfed by mononuclear phagocytes, specifically macrophages. During the inflammation resolution process, these fatigued neutrophils are similarly taken up by macrophages through the mechanism that I described before, which is called efferocytosis. Neutrophil count and function serve as markers for inflammation, with their gradual decline in quantity and the rapidity of their clearance signifying the progression towards inflammation resolution. In the mentioned study, Caravaggio and colleagues administered vagus nerve stimulation 1 hour before intraperitoneal Zemuzan injection and subsequently collected peritoneal exudate samples at intervals of 4, 12, 14, and 48 hours following the injection. The researchers observed that vagus nerve stimulation led to a heightened rate of inflammation resolution compared to control group. Collectively, these findings suggest that inflammation subsided more rapidly in mice injected to vagus nerve stimulation in comparison to the control group. Also, the process of macrophage efferocytosis involving the engulfing of neutrophils was significantly increased in vagus nerve stimulation treated mice at the 12 hours mark following Zemuzan injection as opposed to the control group. Also, the authors conducted in vivo assessment of specialized pro-resolving mediators following vagus nerve stimulation. Specialized pro-resolving mediators are a diverse category of lipid signaling molecules that play a role in resolving inflammation by counteracting pro-inflammatory cytokines and facilitating macrophage efferocytosis. Vagus nerve stimulation treated mice showed a positive correlation with elevated levels of specialized pro-resolving mediators particularly those derived from the omega-3 fatty acids. Also, the researchers demonstrated a decrease in the ratio of prostaglandins, which is pro-inflammatory lipid signaling molecules, to specialized pro-resolving mediators in vagus nerve stimulation treated mice. Collectively, these findings suggest a transition from pro-inflammatory state to a pro-resolving phenotype in mice subjected to vagus nerve stimulation. These results have become the gold standard in the field of bioelectronic medicine, playing a key role in translating preclinical investigations into clinical applications. In recent times, numerous new neuro-immune pathways have come to light with a primary focus on devising interventions that modify the equilibrium between pro-inflammatory and anti-inflammatory cytokines. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.